Hi everyone, and welcome back to our biochemistry series. I'm glad you're here, and today we're going to explore a fascinating transformation that happens to simple sugars inside your body that fold into rings. Yes, that's right. Sugars like glucose and fructose, which are usually drawn in straight chains in most textbooks, don't usually stay like that in real life. In water, inside our cells, they form rings, structures that are more stable, more useful, and absolutely essential in metabolism, DNA, RNA, and even food chemistry. So let me ask you something. Have you ever wondered why most sugars are shown as rings in biology, not chains? And more importantly, how do these rings actually form? That's what we're uncovering today. By the end of this lesson, you'll be able to understand the difference between open chair and ring structures, see and how why sugars like glucose and fructose form cyclic rings, identify the Pyranose and Furanose forms of sugars, learn what an anomeric carbon is, and what A and B anomers mean, and see how these details play a key role in metabolism and biological molecules. So if you've ever struggled to understand why sugars bend and form loops, stay with me. It's simpler than you think, and by the end, you'll be able to visualize it clearly. Before we look at ring formation, let's ground ourselves in what you already know, the structures of monosaccharides, especially glucose and fructose. Think of D-glucose. It's a 6-carbon sugar with an aldehyde group at the top, C1, a series of hydroxyl groups along the chain, and a CH2OH group at the bottom, which is the C6. That's why we call it an aldohexoses, aldo for aldehyde and hexose for 6 carbons. Now take D-fructose, also a 6-carbon sugar, but this one has a ketone group at C2 that makes it a ketohexose. Both are usually drawn in the Fischer projection. That's the flat, vertical form where carbon atoms are aligned vertically and the hydroxyl groups branch off the sides. But here's something to remember. These drawings are 2D simplifications. In reality, sugar chains in water are flexible. They move, bend, and fold, which is what makes ring formation possible. So checkpoint question. If glucose forms a ring, which carbon OH reacts with an aldehyde on C1? Let's zoom in on what happens when glucose is in water. The aldehyde group at C1 is quite reactive. The hydroxyl group on C5 has an oxygen atom with a pair of ion electrons. It's a nucleophile. That oxygen attacks the carbon of the aldehyde and a new bond forms suddenly. C1 and C5 are now connected through oxygen, and just like that, a 6-membered ring is born. The ring contains 5 carbon atoms, C1 through C5, and 1 oxygen atom from the C5 hydroxyl. It's called a pyranose ring because it resembles the structure of a molecule called pyran. The same concept applies to fructose, but there, the reaction happens when the ketone on C2 and the OH group on C5 or C6 forming a Furanose rings, a 5-membered ring. So sugars form rings to stabilize themselves, and this is what they look like inside our body most of the time. Let's give these ring structures a proper name. A pyranose ring has 6 members, 5 carbon, and 1 oxygen. That's what glucose typically forms. A D-glucopyranose. A Furanose ring has 5 members, 4 carbons, and 1 oxygen. Fructose in many biological situations, like in sucrose, prefers the Furanose form, D-fructofuranose. Even ribose, the sugar in RNA, forms a Furanose ring because it only has 5 carbons total. These ring structures are typically drawn as Haworth projections, where the ring is flat and you can easily show whether substituents like OH or CH2OH are pointing above or below the plane. Let's walk through the mechanism slowly. In glucose, the OH on C5 uses its electron to attack the carbonyl carbon at C1. The double bond in the carbonyl breaks and become a single bond. The oxygen picks up a hydrogen, becoming an OH group. Now you've got a new bond from C5 to C1 through an oxygen. Your ring is closed. This new ring is called hemiacetal. For fructose, the same kind of reaction occurs but between C2, the ketone, and the hydroxyl on C5 or C6 that forms a hemiketal. This reaction is intramolecular, meaning it happens within the same molecule. It's like the sugar bite its own tail. When the ring closes, we now have a new kind of carbon to identify, the anomeric carbon. This is the carbon that was once the carbonyl. C1 in glucose and C2 in fructose. Here's where it gets interesting. The new OH group on the anomeric carbon can point either up or down depending on how the ring closes. If the OH is below the ring, opposite side from the CH2OH, it's the A anomer. If the OH is above the ring, same side as CH2OH, it's the B anomer. These forms can interconvert in water, a process called mutarotation, and these small differences can matter a lot. Some enzyme can only recognize A or B, but not both. So I have a question. If the CH2OH group is up and the anomeric OH is also up, is it A or B? Now that glucose is in a ring, let's look at how it actually sits in space. It's not flat like the Howard structure suggests. It falls into a shape called the chair conformation, which is more stable and less crowded. Picture each carbon in the ring as small C. Attached to each are two arms. One sticks mostly up or down, axial, and the other reaches out to the side, equatorial. Bigger group like OH and CH2OH prefer the equatorial spots because there's more room. In BD-glucopyranose, all those bulky groups land in equatorial positions. That's what makes it extra stable. That's also why B-glucose is the dominant forms in water and why it's used to build cellulose, the strong material in plant cells' walls. So the chair isn't just a shape. It's a key to why glucose works the way it does. Let's look at some real-life examples. Glucose in a solution is mostly in the pyranose form. Fructose often forms the Furanose ring in nature, especially in sucrose. Ribose found in RNA always forms a Furanose ring. It's built into your genetic code. Deoxyribose found in DNA is a similar Furanose. Knowing the rings forms helps us predict how sugars interact, how they react, and how they fit into biological molecules. Here's why all of this matters. Glucose is the fuel for cellular respiration. It exists in the pyranose ring inside your cells. Starch and glycogen are long chains of AD-glucopyranose. Cellulose is made of BD-glucopyranose. Our bodies can digest it. RNA and DNA rely on pyranose rings for their sugar, phosphate backbones. Enzymes recognize specific anomers. For instance, lactase only works on B-lactose. In Benedict's test, sugars must open into their chain form, so only those with free hemiacetal reacts. Even the shape of the rings matters. The chair conformations of glucose, for example, places bulky groups in stable position. So let's wrap it up. Monosaccharides like glucose or fructose naturally form rings in solutions. These rings forms pyranose and furanose are not just structural quirks, they are biologically essential. Ring formations happens through intermolecular reactions. The new anomeric carbon determines A or B forms. This form influences enzyme activity, digestions, genetics, and even food tests. Sugars are more than sweet. They are structural, functional, and foundational. Thank you for sticking with me through this journey. I hope you now see sugars in a whole new light. Goodbye!